So today is our discussion for last week's lecture. This will be our last lecture in the series. But still, if you have some issues, you can ask me in the end. The trend will follow as we have been doing from last 11 lectures, 11 or 12 lectures. I'll first give you a recap of what you guys have studied in the last week, and then we will discuss the assignment questions, and you can ask me your queries also, either in between or in the end. That is your choice. Issues on that. Okay, so let's start. Yes. So, in the previous weeks, we have seen what are the tools in molecular biology, how it can be used, what are the different terms that are used in molecular biology, and everything. Now, this lecture mainly focuses on the application of molecular biology. You have done the cloning, you have done each and every process, northern blotting, western blotting. You've learned about cell biology. You know how to do cloning and all of the procedures, but how what are the usage of these procedures, how can you use these procedures in your daily life, or how are these procedures being used in the world. Okay, so today we are going to learn about the applications of the molecular biology. So first of all, in the application series, molecular biology can be used in the field of agriculture, can be used in pisciculture. Pisciculture is actually term used for the fish culturing, and then the poultry, then vaccines, transgenic animal formation, medicines, genetically modified organisms, drug delivery. Many fields are there in which molecular biology can be used one or another way. So there are many applications of molecular biology. First of all, if we'll go but we are not going to discuss everything in detail. We are going to take only four or five things that we will be discussing. First of all is the genetic engineering, how molecular biology helps in genetic engineering. So if you guys remember, we learned about cloning, how we can clone a desired gene in a particular vector, and that procedure is called as the genetic engineering or molecular cloning. So for that, obviously, the molecular biology comes in handy. How to choose the vector, how to digest the vector, which enzymes to use on the insert, how to ligate them, and then further how to transform them in their particular or suitable cells, and then finally screen them to get the desired results. So this is all what we learn about molecular biology in genetic engineering, and that is how the molecular biology is used in genetic engineering in producing a particular type of trait in a particular organism that we want according to our requirements. So that is what genetic engineering is. The procedures are again the isolation of genomic DNA, the polymerase chain reactions, restriction, digestion, then ligation, then competent cells preparation, then transformation, and then screening of clones. That is what is the whole procedure of genetic engineering is. And the tools that are required to do this procedure is the polymerases, the restriction enzymes, the ligases, the vector, the insert, the cells, and obviously the screening agents. What are the methods of screening? Okay. Apart from that, when we have cloned a particular gene in a particular vector, and then transformed in a particular type of cells, then there can be many usages of those things. And for that, in the line of examples, enzymes is the best example that can be given. Enzymes are being used in many kinds of fields nowadays. Food industry, in food industry there are many branches where enzymes are used. So enzymes has a very vast kind of usage nowadays, and the using of these enzymes from a particular type of cells and getting them modified according to our desire, using them in a proper field that we want to use it. Genetic engineering comes in very handy. How it helps is, in case of if I'll talk about dairy production, brewing industry, baking industry, wine and fruit juices production, and meats. So, all of these industries require some kind of enzymes for safekeeping of these products, for processing of these products. So, basically, the enzymes has a very vast usage in food industry, and the thing that makes it possible, the using of these enzymes, is the genetic engineering. So if we'll talk about dairy industry, we have four enzymes that can be used: rennet, lactases, proteases, and catalases. Okay, sorry. So what does rennet do? Sorry. Extracted from the fourth stomach of young calves, it contains an enzyme that causes milk to become cheese. So rennet is used in the production of cheese. It separates solid curds and liquid whey. Whey is the protein that is found in the milk. Different animal rannets are used for different cheese. Depending upon what kind of a rannet is used, there are many kind of cheese that are produced. Most common vegetable rannet is thistle. That rannet is called as thistle. Okay, next is the lactase. What is the usage of lactase? So it is present in the brush border of the small intestine, artificially extracted from the yeast required for the digestion of whole milk. Because the sugar that is present in milk is lactose, so the enzyme required to digest that sugar is lactase. Okay, used in the production of lactose free milk, also used in the production of ice cream and sweetened flavors and condensed milks. So it is used in the production of ice cream industry vastly. Then the catalases, so produced from bovine livers or microbial sources, breaks down hydrogen peroxide to water and molecular oxygen. Along with the glucose oxidase, it is used in treating food wrappers to prevent the oxidation. Basically पैकेजिंग मटेरियल को इस तरह से बनाने के लिए कि वो हमारे खाने को जो है गंदा ना होने दे, बेसिकली खराब ना होने दे जल्दी. So for that, catalases are also used to remove the traces of hydrogen peroxide in the process of cold sterilization, basically to safe keep the food. Okay. Then proteases, so the widely distributed in the biological world, hydrolyzes the specific peptide bond to generate para-K casein and macropeptides in production of cheese. Again, in the production of cheese, these proteases are used along with rennet. Okay, results in the bitter flavor to the cheese and also in the desired texture. Okay, next come the brewing industry. In brewing industry, again the proteases are used here also: beta glucanase, alpha amylase, and amyloglucosidases. So these are the enzymes that are used in the brewing industry. Okay, so where they are used in brewing industry, proteases, if we talk about proteases, works to provide the wort with amino acid nutrients that will be used by the yeast. So basically it is for the production of the products that can be used by the yeast, so that the brewing can happen. Brewing, basically the fermentation part can happen. Okay, so proteases work to break up the large proteins, which enhances the head retention of beer and reduces haze in fully modified malts. These enzymes have done their work during the malting process. So this is basically the brewing industry we are talking about. Beta glucanase represents a group of carbohydrate enzymes which break down glycosidic bonds within the beta glucan, aids in the filtration after mashing and brewing. Okay, so all of these things are involved in the brewing. Then the alpha amylase converts starch to dextrins in producing corn syrup, solubilizes carbohydrates found in barley and other cereals used in brewing, and decreases the time required for mashing. Sulfur amylase, again, a very potent enzyme. Then in the baking industry, if we talk about maltogenic amylase, glucose oxidase, and pentosanase. ठीक है, so these are the enzymes that are used in the baking industry. Maltogenic amylase flour supplement होता है ये. It has an anti-stalling effect, so that the life of that baked item can increase. So it has that anti-stalling effect. It modifies starch while most of the starch starts to gelatinize. Resulting starch granules become more flexible during storage. So basically to increase the shelf life of the baked item. Then the glucose oxidase oxidizes glucose and produces gluconic acid and hydrogen peroxide. H2O2 is a strong oxidizing agent that strengthens the disulfide and non-disulfide cross-links in gluten. Good working condition helps proper function of the bakery system. So again, it helps in the uh, longer shelf life. ठीक है, then the pantocenases, so exact mechanism is still not yet discovered, improves dough machinability, yielding a more flexible, easier to handle dough, because it is the dough in the baking which plays the most crucial role. So for that pantocenases are used. The dough is more stable and gives better oven spring during baking. ठीक है. Then in the wine and the fruit industry, so pectinases and beta glucanases again have been used here. Pectinases prevents pectin from forming haze and hence to get clear solution. Additionally used for the extraction of color and juices from fresh fruits. And it breaks down the pectin and releases methanol, which in high amounts is hazardous. ठीक है. Then the beta glucanase, it accelerates the biological mechanism linked to maturation on lees, reduces the maturation duration, and obviously it helps in the maturation faster. It improves the clarification and filtration and improves the action of fining agents. In basically the wine industry it is used. Then in the meat industry, again the proteases and papain are used. Proteases cleaves the bonds that holds the amino acids together, as enzyme breaks apart proteins, which disrupts or loosens muscle fibers and tenderizes it. Papain is an enzyme which is found in papaya. That is why the pregnant ladies are forbidden to eat this fruit, because this can cause contractions inside their uterus. So again, we are not going into that detail. What is the role of papain here? 95% of meat tenderizers available in the grocery store are made from papain. It is extracted from the latex in the papaya fruit. These enzymes are purified and solved in the liquid form powder or liquid form. ठीक है. Then again, the applications, these were the applications in the food industry. Now comes the thing, the application in the medical science. So what are the applications of molecular biology in medical sciences? Medicines are the class of molecules used to correct the disturbance in the host physiology. They can be chemical in nature, used to inhibit aberrant enzymatic activity from the host or pathogen. In few cases, host enzymes can be supplied as a drug formulation to drive the biochemical reaction. So, biotechnology has potentials in contributing in the development of drug molecules. So basically, it helps in the formation of drug molecules, production of therapeutically important proteins. It can help in gene therapy, we have seen in the previous lectures. It can help in the formation of the vaccines, or it can help in the monoclonal antibody production. All of these are the applications in the medical science for this molecular biology. If we'll talk about therapeutically important proteins, so the large number of genetic or metabolic diseases can be corrected by supplying proteins or factors. ठीक है. So following the, for an example, insulin in the diabetes. Okay. So following the advancement in the biotechnology, many other proteins or factors uh, produced in the different bacterial expression system. In an approach, gene of the enzyme or proteinaceous factor is cloned into the appropriate plasmid to produce the recombinant clone. ठीक है. इसका बहुत अच्छा एग्जांपल है इंसुलिन. Sorry. Next is the production of human insulin, as I told you. So insulin is a dimer of A chain and B chain linked by disulfide bonds composed of 51 amino acids with a molecular weight of 5808. A schematic presentation of the steps involved in the insulin production are given here. In this process, gene A and gene B is cloned into the bacterial plasmid separately to produce two recombinant clones. Then the peptide chain A and B are overexpressed. हम लोगों ने last lecture में देखा था. In the E. coli and recombined together to produce the functional insulin, which can be then further used to provide to the patients of diabetes in which the insulin levels are low. ठीक है. Another proteins that can be important therapeutically are the, sorry, factor 8 and factor 9, which are important for the hemophilia patients because these help in the blood clotting reaction. Tissue plasminogen activator, lactoferrin, human protein C, alpha 1 antitrypsin, fibrinogen, pro 542, antithrombin 3, collagen 1, and serum albumin. So, all of these proteins are important therapeutically, which can treat one or other disease. ठीक है? So, again, we are not going into the detail of the recombinant chymosin or human growth hormone and blood clotting factors because I've told you these are important. Next is the application in the gene therapy. So, production and supply of recombinant protein is a temporary solution for the treatment of a disease. Obviously, ठीक है. In another approach, human expression system is used to produce the proteinaceous factor after inserting the recombinant clone into the human cells or inside the human body. Recombinant DNA is packed into the appropriate DNA delivery system to deliver the gene into the human cells to correct the mutated genes or encode a therapeutic protein drug to provide treatment. So basically, a gene which is faulty in humans and for which they need to take supplements from outside. So instead of taking the supplements, if we can clone that gene in a cell where it can divide and overcome the situation that has happened due to the lack of that particular gene, so that is what we call gene therapy. ठीक है. There are two types of gene therapy, somatic gene therapy or germline gene therapy. Therapy, in this therapeutic approach, the therapeutic genes is transferred into the somatic cells as per the requirement of the individual to treat the functional defects, and this treatment does not move to parents, offsprings or next generation because it is somatic gene. While in case of the germline gene therapy, in this therapeutic approach, germ cells are transformed by the introduction of the required gene to produce the protein or correct the mutated gene. So it is in the germline, that means it will be transferred to zygote, and if it will be transferred to zygote, that means it will run to next generations also. So germline gene therapy is for that. Okay, next is a vaccine. It is given to develop immunity against the disease in the human or other vertebrate animals. Vaccines are dead attenuated organisms or protein derived from them. They are different strategies to enhance the immunobiological response to give long lasting protection against the disease with minimum adverse effects can be seen in case of the vaccine. ठीक है. Again, vaccines can be of different type depending upon what part of the organism is involved in formation of the vaccine. It can be heat killed, it can be attenuated, it can be toxoid, it can be subunit, or it can be a conjugate. ठीक है. So there are many types of vaccines present. Then comes the thing that is the application in the transgenic animals. So basically, what are the transgenic animals? Transgenic animals are those animals in which a gene has been transcribed for our interest, and then the animal will produce that particular type of protein for us. ठीक है. Generally for organ transplant kind of expressions, these transgenic animals are produced. So ठीक है. So overexpression, transgenic animals can be either overexpression of a particular gene or it can be a knockout. ठीक है. So overexpression means expression of gene into suitable form used for the organ transplant. Knockouts are the removal of a gene expressing the functional gene useful for understanding the role of that particular gene. That means what will be the changes inside the animal in the absence of that particular gene. ठीक है. So understanding the disease mechanism, disease models, conventional methods, naturally occurring genetic mutations, chemically induced disease conditions, streptozotocin induced disease models. सो बेसिकली लिमिटेशन होती है ट्रांसजेनिक एनिमल के साथ कि नो प्रोसाइज कंट्रोल, ग्रॉस चेंजेस इन द फिजियोलॉजी, contribution of multiple organs etc is required in the transgenic animals. ठीक है. So these genetically altered animals will have a resemblance with the signature pattern of the human disease that we want to treat, that will be copied in the organism which is in the animal which is transgenized, so that we can understand ki what kind of a treatment will work on human or what kind of a thing can overcome the disease. ठीक है. So major advantages, target specific genes होती है, no human subjects, cheap and quick होता है and reproducible होता है. ठीक है, although it is also very difficult, but yes. Next is the AIDS, that is acquired immunodeficiency syndrome model. So to understand this disease, this kind of a model was developed in animals. So monkey can get infected with SIV, not AIDS. So first AIDS mouse model, SCID mouse, and other animals used for AIDS model are rats, cats, rabbits, and Drosophila. And advantages क्या हैं इनके? They are cheap, they are much is already known about their genes and everything. Okay. Now the Alzheimer's mouse. Alzheimer's is one of the most prevalent brain disease in India. So for that, to test what kind of effect this disease causes on brain or what kind of treatment can work on humans, this Alzheimer mouse models are prepared. It is a neurodegenerative brain disorder, amyloid plaques, fragment of beta amyloid precursor proteins, APP mutation to induce peptide aggregation in the key to develop Alzheimer mouse models. ठीक है. And of which the strain which is a very good transgenic mouse model to support with to start with the order to produce genuine human amyloids is TG2576. First Alzheimer mouse was incorporated an APP mutation. ठीक है. APP mutations basically amyloid beta peptide beta amyloid precursor protein. ठीक है. This kind of a mutation was observed so that the Alzheimer's can happen in the mice and then we can study. ठीक है. Then the cancer mouse models are possessed to study cancer formation to screen anti-tumor drugs. Oncogenes are either mutated to induce spontaneous cancer formation, knockout of the tumor suppressor gene. HER2 overexpression is associated with the cancer formation. HER2 is a gene involved in the cancer and breast cancer, and 13 different strains were engineered for the production of this cancer mouse model. ठीक है. एप्लीकेशन हम लोगों ने देखी है टू स्टडी डिफरेंट डिजीजेस एंड ऑल. The transgenic animals are also used for the organ transplantation in some cases, but obviously they need to match many factors for that so that body don't reject the transplant. ठीक है. So advantages of transgenic animals क्या है? Gene requires certain cellular mechanism to help for the production of protein. The animals used for transgenic purpose naturally carry the mechanism needed to produce complex proteins. These mechanisms is absent in cell culture. Expression through cell culture or bacterial culture requires constant monitoring and sampling. The isolation and purification of expressed protein is conventional method is more difficult than purifying proteins from an animal's milk or body fluid. So there are many advantages of transgenic animals, while the disadvantages are also there, but advantages are also there. Then the application of PCR. So PCR is used for first amplification of your gene of interest, molecular identification के लिए use होता है, sequencing के लिए use होता है. For if you remember, we learned about Sanger sequencing, Maxim Gilbert sequencing and all, and genetic engineering obviously for molecular cloning. ठीक है. So we are again not going into the detail of the applications of PCR in food microbiology. PCR in medical sciences. So PCR in medical sciences already began in 1985, and a viable test for measuring the amount of HIV in the blood was published in May 1987 by John Sinsky at CETUS. ठीक है. So in medical sciences also it helps in identifying the particular organism. ठीक है. इन्फेक्शियस डिजीज में पीसीआर का क्या रोल है होता है? टेक्नोलॉजी हैज बिकम द बेसिस ऑफ फॉर अ ब्रॉड स्पेक्ट्रम ऑफ द clinical diagnostic tests for various infectious agent including viruses and bacteria. Unlike the traditional antibody based diagnostic tool that depends on the patient to develop antibodies over time. PCR allows faster diagnosis, which fastens the treatment and recovery. बिकॉज ये तो आपको उसी टाइम जीन एम्पलीफाई करके बता देगा इफ द ऑर्गेनिज्म इज देयर और नॉट. Besides detecting the presence of pathogen, PCR allows us to quantify the amount of pathogen present in patients' blood, and this helps to monitor the progression of the infection or response to the treatment. ठीक है, so PCR is again very important in case of the infectious diseases also. It is also used in blood screening. It is PCR helps in detecting the presence of infectious disease in a donatory blood sample. Benefits of using PCR technology to monitor the world's blood supply include a decrease in the waiting period during which the infectious agent is undetectable by serological screening technologies that rely on the formation of antibodies. So again, it is very important in blood screening. ठीक है? Just a moment. Then the plant microbe interaction, the RT-PCR technique, real-time PCR technique, due to its rapidity and sensitivity, has also seen its usage in detection of microbes infesting a crop plant. Early diagnosis of the pathogenesis is essential to provide rapid and suitable measurements for limiting the epidemics and selection of the appropriate control measures. ठीक है, so that is how the post microbe interaction also helps to understand the particular proteins or particular type of organism. ठीक है, in veterinary parasitology also it is used for the disease Ejuski disease. This is a virus of pig, and this virus can cause abortion and mortality in piglets. This disease has a latent period where there is no symptoms of infection, making it difficult to eradicate the disease completely. For this reason, PCR is considered to be appropriate tool for detecting latent classes of Ejuski disease, and PCR assay was developed in 1989. ठीक है, so PCR again is very helpful in detecting the veterinary parasitology also. ठीक है, there are other things also, the BLV, BV, DV. These kind of viruses are also uh, identified using the PCR technique. ठीक है, then in forensic science, the criminal investigation, so PCR is used to basically read the tandem repeats in the particular organism to understand the relation between the different suspects, and then this is how the PCR helps in the forensic science. In case of research, we have been learning from last few weeks. It is used in the DNA cloning, DNA sequencing, sequence tagged sites, phylogeny analysis, and gene expression studies. It is used everywhere. Then the applications of real-time PCR. Real-time PCR is gene expression analysis, drug resistance, disease diagnostics and management, viral quantification, food testing, GMO food, genetically modified food, gene copy number, and cancer research. So all of these are the applications of the real-time PCR. Major application, then we have another thing, that is the zinc finger. uh, 2 minute दिखाया है मैंने. So zinc finger nucleases are there, which helps in the again gene knockout or gene knock-in, depending upon their ability. They can also be exploited, which is again one of the very major uh, application, we can say, from the molecular biology. ठीक है, so it can cause targeted chromosomal deletion. They can modify the genome. They help in crop improvement, application in HIV treatment. ठीक है, gene knockout or gene knock-in. Knockout involves the inactivation of genes or gene segment, resulting in the loss of gene function. This is typically achieved by introducing random insertions or deletions into the genome through the imprecise DNA repair process of non-homologous end joining triggered by double-stranded breaks. Before the advent of CRISPR-Cas9, researcher community relied on random mutagenesis methods such as ENU mutagenesis. induced by the radiation or chemicals. So UV light can also cause mutations, so you should prevent yourself from UV light. Okay? For generating DNA changes, this approach, often employed in zebrafish following forward screening, was less precise. ठीक है, तो knockout is basically the inactivation of gene or gene segment, while the knock-in operates differently by introducing specific base pair changes with precision into the genome. This involves a targeted introduction of the template, क्योंकि यहाँ पे हम लोग नॉक इन कर रहे हैं, बेसिकली एक नई जीन को डाल रहे हैं. ठीक है, एंड दैट जीन विल बी एम्पलीफाइड इन द ऑर्गेनिज्म एंड देन दैट कैन बी एक्सप्रेस अकॉर्डिंग टू आवर रिक्वायरमेंट. ठीक है. So zinc finger nucleases have been used to create targeted mutation in the genome of various organisms. These engineered nucleases consist of zinc finger proteins, which can be designed to recognize specific DNA sequence, and a nuclease domain that induces double strand breaks at the targeted site. ठीक है, so this is how zinc finger nucleases can be used. Deletions can be done in the human cells, and then last but not next is the talent. So if you guys remember, we learnt about ZFN talents and CRISPR Cas9 in some previous lectures. So here is just an application of talents. So customized ZFN and talents have revolutionized the genetic research by efficiently introducing targeted alterations in various organisms and cell types. This has surprised interest in using these technologies for therapeutic purposes in genetic based diseases. Okay. So talents has also many applications in the molecular biology, again to silence a gene or to knock out or knock in a gene. Okay. So this was all about lecture 12. We didn't went into the much details because that was not the requirement. It was just you guys should know what all other areas are where the molecular biology can be applied and can be used. Now we'll discuss the questions of assignment. Meanwhile, if anyone wants to ask any question, they can. Otherwise, we'll start with the question answer round of assignment. And obviously, as we have been doing in all of these lectures, you will type your answer in the chat box. No one will speak up the answer. Okay. Hmm. ये ये छूट गई रे. That's okay. So we'll discuss the questions here one by one. The question number one is dash hydrolyzes the specific peptide bond to generate para K casein and macropeptides in the production of cheese. So what is that enzyme that is used? ठीक है. Question number one, we are talking about what is that enzyme that is used here. The answer to question number one is proteases. ठीक है, आंसर टू क्वेश्चन नंबर वन इज प्रोटीएजेस. आई होप यू ऑल वर एबल टू decode that. Next is the maltogenic amylase results in starch granules becoming dash during storage. More more flexible. So answer to question number 2 is more flexible. ठीक है. Next is the H2O2 is a strong oxidizing agent that strengthens the dash and non-disulfide crosslinks in gluten. So answer to question number 3 is disulfide bonds. ठीक है, it is the disulfide bonds. uh, H2O2 is a strong oxidization that strengthens the disulfide bond and the non-disulfide crosslinks in gluten so that it does not get oxidized. ठीक है, next question is the proteases. Protease enzyme breaks apart proteins which disrupts or loosens and tenderizes the meat. So answer to question number 4 is the muscle fibers. ठीक है, it loosens the muscle fibers and tenderizes the meat. That is what proteases do in meat. ठीक है, question number 5 is insulin is a dimer of A chain and a B chain linked by which bonds? It is again the answer disulfide bonds. ठीक है, the both of the chains of insulin A and B chain are linked to each other by disulfide bonds. ठीक है, next question. In conjugate vaccine preparation, the bacterial coat is tagged with dash to induce production of an immune response against the bacterial coat. In the conjugate vaccine preparation, the bacterial coat is tagged with dash to induce production of an immune response against the bacterial coat. What is that? Yes, it is the immunogenic protein. It is an immunogenic protein. Next question. APP mutation to induce peptide aggregation is the key to develop dash in mouse models. Answer is Alzheimer's disease. It is an Alzheimer's disease. ठीक है, amyloid proteases जो यहाँ पे होते हैं. And next is the dash are either mutated to induce spontaneous cancer formation. What are that? What can be that? The answer to question number 8 is oncogenes, because oncogenes are the genes that are cancer causing genes. Okay. Next is dash acts as a glucose sensor in pancreatic beta cells and plays a central role in the models of insulin secretion. So what is that? Question number 9 ka answer is glucokinase. It is glucokinase. Question number 10. Dash are used to manage bleeding disorders by replacing missing or defective proteins. So what are those? अगर हम हीमोफीलिया की बात करें तो वहां क्या प्रॉब्लम होती है कि ब्लड जो है वो कोएगुलेट नहीं हो पाता. ठीक है, सो उन पेशेंट्स की ट्रीटमेंट के लिए बेसिकली ब्लीडिंग डिसऑर्डर्स की ट्रीटमेंट के लिए व्हाट कैन बी यूज? इट इज द coagulating factors that can be used. ठीक है, it is the coagulating factors that can be used. ठीक. Next question is dash is added to infant formula to mimic the immunological benefits of the breast milk. What are those? So answer to question number 11 is lactoferrin. Lactoferrin provides those immunological benefits. Dash heart valves has been successfully transplanted into the human heart to replace damaged valves. What are those? So question number 12 का आंसर is porcine. ठीक है, it is porcine. Question number 13. The doggy mouse is genetically modified to overexpress dash transgene in the cortex, hippocampus, thalamus, brainstem, and cerebellum. What is that? Question number 13 का answer, everyone. Someone was there. Was there. So answer to question number 13 is NR2B. It is NR2B. Next question is bovine leukemia virus causes dash bovine leukosis in cattle. So answer to question number 14 is enzootic. Enzootic. ठीक है? Question number 15. DNA fingerprinting is using dash loci because the loci are so variable that unrelated individuals are unlikely to have the same variable number of tandem repeats. ठीक है? VNTR means variable number of tandem repeats. So answer to this is again VNTRs, that is variable number of tandem repeats. ठीक? VNTRs. These are actually used to do the DNA fingerprinting. So variable number of tandem repeats are never same in two individuals. They are only similar to their parents. ठीक? So this was all about week 12's lecture. If anyone has any confusion, they can ask me. And this is what we have started in the last week's lecture. Still, if you have any issues, you can ask me. And I hope you guys do well in your exams. All the very best for your exams, and all the very best, everyone. So I'm waiting. If anyone has any query, they can ask me. We've seen how molecular biology has so vast applications. It is nearly involved in every kind of industry, be it food industry, be it medical field, be it any industry that is involved that involves the living organisms. This molecular biology has applications in. So it is a very interesting field to study about because it gives you so much to explore and so much to perform, even in the field of research also. So this is a very interesting field to study about. I hope you guys enjoyed the course. हम्म। ये क्या हुआ वाईफाई? Gene therapy is again a very important field to study about, because this is something which is used in the upcoming world. If you guys have heard about the baby designing thing, then that is also something related. Not exactly gene therapy, but that is also something related to gene therapy. So it is a very upcoming world thing that you guys should study about if you want to get indulged into the molecular biology. Okay, so this was what there in the detail about molecular biology as sir has taught, and I hope you guys understood it. Mm, mouse models, this cancer mouse model is again they are performed on the nude mice. Also, nude mice has almost nil immunity in them, so they are very delicate to handle. They are very, 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 delicate and very prone to disease kind of animals, so they must be handled very carefully. And HER2, as I told you, is a gene associated with the breast cancer, which is again uh, used to cause some kind of a cancer in case of mice model, so that can be studied further for the testing of the cancer. Actually, we didn't discuss about the transgenic animals disadvantages. So the transgenic animals are, again, it is a very expensive process to make a transgenic animals. Generation of transgenic animals are all because they are expensive because the gestation period is long, litter size, and high maintenance cost of the recipient animals. There may be high mortality rate and other deleterious effects on animals used by the researchers to create the transgenic breeds. It has been observed that transgenic pigs having enhanced growth rate and efficient feed conversions exhibit reduced reproductive performance and may suffer from arthritis and dermatitis, etc. Large number of recipients is required for embryo transfer because of the low transgenesis rate. And transgenic foods have been produced and offer better productivity in terms of both yield and quantity, but obviously there is some kind of compromise on the quality, or it may be an allergen to some people. So yes, transgenic uh, animals producing of transgenic animal has its own disadvantages also. It is not like we can go ahead with the field just like that. There are disadvantages for this also. So, in infectious diseases, the PCR helps in a way. It helps in the monitoring of the progression of the infection. So, for example, for HIV, as it was used in case of the SARS coronavirus 2 also. So, in that case, also PCR helped a lot. It is a very well known example. Apart from that, hepatitis B and C viruses, human papilloma viruses, chlamydia trachomatis, which results in the infertility in women, Neisseria gonorrhea, which is again an STI, causes the pelvic inflammatory disease in women, and cytomegalovirus, which affects more of the transplant patients and immunocompromised people such as AIDS patient. ठीक है. So yes, there are many kind of infections that can develop, and PCR helps in detecting those kind of infections. So I hope you guys understood the course well, understood the lectures well. If you have any confusion, you can still contact us. Thank you so much for joining. Bye, everyone.